Hello, folks. Hope you all are doing well. Sorry that we cannot post a video for a longer period in time. So I would appreciate the patience showed by our subscriber. So thanks a lot. I was very busy with the scientific work that I'm on to, but I'm just trying to come up with a particular video, so which is very short and crisp and describes about the things which are mostly in use now and then you are listening about the same, that is your AI/ML based tools. Both are AI/ML based tool. One is AlphaFold, as from the thumbnail you must have got the idea what we are going to discuss today. So that is your AlphaFold, which is used for protein modeling. So I have also described the particular tool beforehand in the first video. But we'll also be looking at Alphamissense. Okay, so both the tools are from this company called as DeepMind. Okay, so Google's DeepMind, because DeepMind is a UK based company which is acquired by US based company Google. So now it is Google's DeepMind, and as you know, AlphaFold being the Nobel Prize winning software, or basically the team has got the Nobel Prize for the discovery of this particular AI tool. The reason I will be telling you in a while. Why is it so much revered, and why this particular tool is highly rated? Okay, for protein modeling, because it can reach a similarity of up to 98%, basically 98 99% of similarity to the wild type structure which is procured using wet lab experiments. Okay, so via the computational means and via the computational predictions, if you can reach to the precision of 98 99%, so that's a great thing, because in the wet lab experimental techniques like your NMR, cryo electron microscopy, and this that, so that would be taking a whole lot of time to procure, like 4 5 years or so. Okay, whereas this particular thing you can do in 2 to 5 minutes only. Okay, and if you can reach to that particular level of precision, of course it goes without saying that it's a very good tool. Okay, so how it is able to do that, so we'll be talking about those lines. Okay, so a brief on that, and then we'll be talking about Alphamissense. Okay, so Alphamissense has a beauty that it can do the prediction of variances, okay, which are not yet been discovered even. Okay, so because it is a AI based tool, so it can predict what sort of a mutation, which at every position what particular mutation or the alteration of amino acid can lead to what sort of a difference. Okay, so basically the USP of this particular tool is that it can predict the variance at proteomic level. Okay, whereas we have plethora of tools like ClinVar, ClinGen, dbSNP, and there's that dbGap, dbVar. Okay, so which can analyze the variance, or dbSNP basically. So basically those are the tools which can predict variance at the genomic level. So that is the first thing. Second thing, it can only show you the data which is already being like fed onto it. Okay, so it is not doing any sort of prediction, and it is not a AI based tool like all of the NCBI based tools are not AI based tool. So it just gives you the data. It is just based on the search that you get. Whereas Alphamissense is trained using ClinVar data, as well as it utilizes your alpha fold. Okay, so alpha fold for your protein knowledge basically, and to get the variance knowledge, it utilizes ClinVar data, that how clinically relevant that particular variance is. That particular variance might lead to a typical disorder based condition, so that we call as pathogenic, or like it is benign in nature. Basically there are very less chances of that particular variance to be causing a disorder, yes or no, likewise. Okay, so let's get started without further ado. So we'll be talking about AlphaFold at first. Okay, so I would encourage to go through this particular paper if you are very new to the subject. Okay, so you go through this particular paper. Okay, so although this is not the paper, so this is the site that we're talking about. This is the paper published in Nature. Okay, so let's go on to the paper once before jumping onto the web server. Okay, so this particular web server you can use to search for the proteins, but if you want to predict the structure, you might go to the yeah. So basically it is hosted by NVIDIA. Okay, so NVIDIA based server we are talking about. So I've demonstrated the particular server, like you can utilize bolts to ESM fold and Alphafold in there as well. Okay, so using that particular server, I will give you a glimpse in this particular video also for the new subscribers or for the new audience which who is listening to this. Okay, so coming back to the session, like this particular demonstration, basically when we talk about, so there you can go through this particular paper, it has multiple of the things, but just I need to explain this particular thing which is very important. Okay, so here you can see the blue one is basically AlphaFold predicted structure, whereas green one is the experiment based structure. Okay, so experimentally procured structure. So it can be by the use of X-ray crystallography, NMR, or cryo-electron microscopy, electron microscopy, and likewise. Okay, so that would be the experimental structure. Okay, whereas like the blue one is predicted using AlphaFold, and you can see there is a higher precision in this one. So okay, so if we do a superimposition, okay, so if we are basically superimposing, we can see that all of the contours or all of the coils or helices, okay, everything is following over here. Okay, so this much of a precision you can get only in 5 minutes to compare to that of experimental structure which takes 4, 5, 6 years maybe to procure. Okay, that much of a precision, not only at the periphery of it, but also you can see at the active site, or like if we are trying to look at the like site specific, or basically we are looking at the domain specific things, still it can predict those particular things also with higher precision. Okay, so here you can see it is like almost superimposing 88%, like 98% or likewise. Okay, and the whole protein structure, so this is one of the chain or the helix base N terminal C terminus we are talking about, and this is the whole holistic overview of the protein. Here also you can see the overlap. Okay, and here also you can see the domain specific also you can see. So RMSD, the lesser it is, better it is considered, because RMSD concerns for like what is the initial state of the whole protein to the final state of the whole protein, or like whichever experiment we are doing. So there should not be much of a change in the protein structure, or the integrity should be maintained, and the lower this particular number is, better it is. So here you can see 0.8 is a good number, anything below 2 angstrom is considered mostly good. Okay, so but this is for the whole protein. 2.2 angstrom is also a very good what do you say, your score. Okay, because for the whole protein. So here this is the workflow of the things that are going in the background when we talk about AlphaFold. Like it is doing your multiple sequence alignment search, alongside that it is also doing the pairing thing and simultaneously looking for the templates. Okay, and the main thing which makes this AlphaFold different from all of the tools out there is this particular Evo former. Okay, so Evo former is the main thing which is the highlight, and which is basically making the things worthy enough, because it can do parallel processing of all of the things which it has to look upon. Okay, apart from that, it can also predict structures of the protein which are not yet been deciphered in the experimental setup. So that is the main thing. So basically, just a trivial fact that all of the protein known to the humankind as of now is being modeled using AlphaFold. That much of a powerful tool it is. Okay, so if you go to your PDB, protein data bank, you would find that there are more than 10.5 lakh of protein that are being shown over there, which are done using computational modeling, and most of them are using AlphaFold only. Okay, so the raw data that you get like is in the form of PDB, either via experimental means or by computational means, or you have your AlphaFold based structure, which is totally computational thing. Okay, UniProt and all are third party tools which will cater, or basically will be having structures from all over, structures from AlphaFold as well as PDB, everything. So it is a third party tool. It does not have anything of its own, so it will incorporate all of the all of the information about a protein in a particular place, so that it is a one-way stop for getting all of the information. Okay, so that is about it. So I can also show you, once we go on to the PDB, you can get to this particular thing what I'm trying to make a point of. Okay, So if you go to PDB, you can see at the top left basically that you have like how many of the experimental structures you are having, how many computational structures you are having, this one. Okay, so your experimental structures are about 2.5 lakh, whereas computationally modeled structures are more than 10.5 lakh. Okay, so 5 times of this one, because as I have already stated, all of the protein known to the humankind are being modeled using AlphaFold. Okay, and like very fast. Okay, so that is why it is of so much of use. Okay, so now getting back to the AlphaFold, so what is the server for that and how to interpret excuse me, which part of the protein is having more precision, or how we can count upon the protein structure that is being predicted. So these are the search terms that you may use. Of course, by the name of the protein, you can search by the gene ID. Basically, gene will be coding for that particular protein. So you can go by the gene ID. Okay, so like BRCA1 or like GATA3 or this, that, or PNPLA3. So those are the gene names. So of course, it would be coding for a protein. So that particular protein would be shown to you. Okay. Suppose that you have your UniProt accession ID that you can give. Okay, all sort of things it will support, even the amino acid structure if you can, or basically amino acid sequence if you can give, so that is also feasible. The examples are also given over here. So let's get started with the sequence of amino acid basically in its FASTA format. Okay, so you don't take the whole FASTA format, you can just take the sequence. Okay, so this is the amino acid sequence here, you can see. Okay, single letter codes we are talking about. So if we go inside this, okay, so it will show me a structure. Okay, so here is the structure. Okay, so this is the structure, and now you can follow the color connotation. What does this particular color represent? So this color is what I'm going to explain right now. Okay, so here you can see it is already written. Okay, so anything in predicted value will always lie, because it is a probability based value, so it will always lie between 0 to 1, as you understand. Okay, so if we convert it to percentage, of course you like do a multiplication of that particular thing into 100, so it becomes a percentage. So basically if I consider, okay, so what is the highest confidence? So very high confidence. So this particular color basically represents the confidence of the protein. Okay, excuse me. So confidence basically means that this particular deep blue color that you have, okay, is having a PLDDT. Okay, so what is a PLDDT? PLDDT stands for pre-residue measurement of local confidence. Okay, so basically in simple words, it is just a confidence based parameter. Okay, so PLDDT, the deep blue concerns with a PLDDT of more than 90. More than 90 means more than 90% of confidence. Okay, it is having. Then the lighter blue part is having a confidence between 70% to 90%, whereas the yellow ones is having 70% to 90%, 50% to 70% confidence. And the yellow one, okay, so the side chains mostly will be having the yellow and orange, and that is good because the side chains does not add up to the function mostly. Okay, the backbone of the protein is more important, so that is why the backbone should be higher on the side of your like confidence score, like the deeper blue or the lighter blue. That would be good. And the last but not the least is your orange color, which concerns with less than 50% of confidence. Okay, so what does the confidence stand for? Okay, first of all, so basically confidence is simple. Okay, just like how you train an AI, okay, just like how you train your brain. Okay, so if I ask you a question, how much confident you are to answer that particular question? Okay, based on your previous knowledge, of course. Okay, if you are doing something, how much confident you are that the answer is right. Just like that, this particular confidence refers to how much of a resemblance it can have with the wild type structure if it is procured experimentally. So let's suppose this particular protein, if I superimpose this particular protein structure modeled using AlphaFold 2 to the experimentally validated structure or experimentally procured structure. Okay, so this particular area, deeper blue area that you see, it will have more than 90% of confidence, means more than 90% it will resemble with the wild type structure. Okay, more than 90% of chances are there. It can be 90 to 100%, that means. Okay, so anything in between 90 to 100% it would be having a resemblance, so that is the highest. Okay, then the lighter blue area might not superimpose exactly, so that would be between 70 to 90% of a similarity you can find with the wild type structure. Likewise, 50 to 70% for the yellow part, and less than 50% for the orange part. Okay, so we have to count more upon the higher values, of course, that would be better. Okay, so that is there. Domain specific also you can go by. Okay, so based on the domain, if you want to color, so here you can see based on the domain you have the colors, color connotation as your green and orange that is being shown over here. Okay, so that is it. Okay, apart from that you can use the Alphamissense also alongside in here, by the way. Okay, at times you can find the, so these are the allowed and disallowed reason. Likewise, okay, so what is the residue alignment. Okay, so everything you can read in here, so all of the information is provided. Apart from that you can also look for the variances. Okay, so if I point my cursor to any of the amino acids, so that particular thing would be highlighted over here. At the bottom you can see what is the PLDDT score of a particular amino acid sequence structure. Here you can see that I have pointed out that is V. Okay, so the position is written. Okay, so which model. Okay, and the PLDDT is 94.75, that is very high. Okay, that is why the part is blue. If I go to model confidence, based on that you can understand much better. Okay, yeah, likewise. Okay, so protein similarity you can go on to then, like how many similar proteins of this particular nature exist. Okay, annotations of this that you can see. Okay, apart from that Alphamissense can also be used alongside this. Okay, so what is Alphamissense, that I am coming on to in a while. Okay, as I have already told you that Alphamissense is used for variance analysis at proteomic level. Okay, mind you that at proteomic level, not at genomic level, because it is far easier to understand the variance at the genomic level. But when the product is done, okay, so it is a final furnished product, that is protein. After you have the furnished product, then if you are able to find the variance and what would be the repercussions of that, so that is a novel thing. That is why the Alphamissense is so much revered. Okay, prior to getting onto the Alphamissense, let me show you the site from where you can actually model your protein, or basically use Alphafold directly to model, because here you can't model, here you can only search for all of the protein that are being modeled using Alphafold. Now if we can do a like the same Alphafold, we can write NVIDIA in front of that, so it will redirect me to the server. Okay, so this is the server Alphafold 2 model by DeepMind from NVIDIA. So they are having the supercomputers in the background. Okay, so here you can see, here they have used the confidence score over here. The color is a bit different here. The higher yellow concerns to higher PLDDT score. Okay, apart from that you can like paste your sequence. Okay, and likewise you can do the modeling. You can hit on run, and based on that you can do the modeling. Okay, so if you don't want to include or you want to include a particular type of sequence, that you may also do, or you can maneuver the shell script as per your liking, or the Python API code. Mostly in case of doing it from end to end, you would need a API key. Okay, so that is just the thing that you would be requiring. Okay, apart from that, whichever are being modeled, so you can see those. Okay, but this is how it works. Okay, apart from that, as I have already told you, you can go to explore and to the biology. Here you will be having so many of the protein modeling tools like AlphaFold 3. Now it is downloadable. Okay, so AlphaFold 3 is there, then you have Bolts 2 is there. Okay, so MSAESM fold. Okay, so this particular video is already there, like the very first video of this particular channel is on this particular Bolts 2 and ESM fold and all. Okay, so you can follow that also. So I'm not repeating or iterating upon the same, that is why. Okay, so now coming back to our Alphamissense, for instance. Okay, Alphamissense. So you can also do a project in these particular things. If you are interested in the same, you can just let me know. So we also do project on these particular things, so those are of your 6 month to 1 year, so it would be a far detailed thing, so where we'll take a disorder and based on that it would be a very very streamlined thing to do. Okay, so then alpha missions, how do we utilize all of these tools at once and to come up with a workflow, and how the results are making sense, because as of now whatever I'm telling you are just tools. Okay, how to utilize those particular tools to come up with a particular result. So if you are in your bachelor's master's, so you can reach out to me for your projects and all. Okay, so we also do those particular things. Okay, so yeah, so there would be a fee concerning to that, so like all of the details you can get from me. Okay, so you can reach me via the LinkedIn profile that are being pasted in the YouTube section. You can go via that. Okay, so accurate proteome wide missense variance effector prediction using alpha missense, that is what I was telling you, that it is a variance effector prediction that you are doing at the proteomic level. Okay, so again I will just be explaining you very fast that how it is being done. So first is structural context is being taken from, so alpha missense has to be trained because it is a AI ML based tool, so it has to be trained first. Okay, and for the training, what is the purpose that it is serving? It is trying to come up with the variances at proteomic level, which is very tough as I have already told you. But how do you do that? Basically the DNA or the gene is basically coding for that particular protein. Okay, so the coding part of the gene, of course we are talking about the exomes. Okay, so or exons that are basically contributing to the like a coding part of the gene, and that would be coding for a particular protein. Of course we are not talking about the silent ones and likewise. Okay, so basically when we have the protein, so there are two things that we require to train our machine learning algorithm using, so that it is able to predict the structure and also predict that which particular amino acid change might bring what sort of a functional changes, and that will basically infer that the variance will cause a disorder, how much of a probability is there to cause a disorder, and how much devastation it can bring basically. Okay, so pathogenicity basically. So for that particular context, we have to train it using the protein knowledge because it is predicting the structure. So or basically it would be requiring the structural knowledge. For that we have to train it using Alphafold. Okay, so Alphafold is from the same company, that is why it is far easier for them to train it, because the alpha missense is also from DeepMind, Alphafold is also from DeepMind, so that is why. Okay, so it is an internal thing, so they have used Alphafold for the training purpose, and from where it is taking the structural context, how the amino acids are gonna fold, and the protein knowledge of it as well. Okay, so how it is gonna fold and what would be the template, everything it is getting from the Alphafold, and it is getting the structure, integrity, everything it is getting, how the amino acid is gonna fold in 3D and everything, but it would also require a variance knowledge. And for the variance knowledge, it has to go to a like web server which has that particular knowledge, that how clinically relevant a particular variance is. Clinically relevant means which particular variance is gonna cause a disorder, which is not gonna cause a disorder, which will be benign type, which will be a pathogenic type, everything. Okay, so benign type basically means very less chances of getting onto the disorder or disease state, whereas a pathogenic has the highest probability of being converted to a life-threatening condition, or basically any of the clinically relevant condition. Okay, so likewise, so based on these two type of training, so that particular data will be taken from ClinVar, because ClinVar has that particular data, clinical variance. ClinVar from NCBI, you understand. So it is trained using Alphafold as well as ClinVar. Okay, Alphafold for the structural context and protein language modeling, whereas ClinVar it is trained using to get the knowledge of the variance and how relevant that particular variance can be in the clinical settings. Okay, because at the end of the day, what is our aim, that which particular variance can land up to a diseased or disordered condition, so that would give us a very good idea in the medical context. Okay, so here you can see alpha missense based pathogenicity screening if you are doing. So the screening is based on benign, uncertain, and pathogenic. In ClinVar, if you go on to, so it would be likely benign, benign, uncertain, likely pathogenic, and pathogenic. So five categories. Whereas in Alphamissense, you only have three categories. Okay, so all the possible variances in this world, 71 million possible missense variances are there. So it also only counts for missense variants, by the way. Okay, not for other type of mutations. Okay, so there are plethora of different types of mutation. It only counts for the missense variations. Okay, because those are clinically highly relevant. That is why. Okay, so change in the amino acid and how that particular change in the amino acid can bring a change in the further protein structure, and the structure concerns to the functioning of the protein, that like basically changing the functionality of that particular protein, and that may hinder the further cascade of reaction in the metabolome. Okay, so metabolomics that you follow, so in the metabolomic analysis or in the metabolite formation, if there is a like malfunctioning, so that will cause devastation, like your most of the disorders would be having a like metabolic disorders like your diabetes and this that, the insulin is not produced well because of the islet cell from your pancreas are not working, and likewise, so there are multiple of the hormonal changes and this that you can expect. Okay, so basically out of the 71 million possible missense variants, 57 are likely benign, not very likely to cause a disorder. 11% we are uncertain about it, 50-50 basically. Whereas 32% of them are pathogenic. So that means those particular variants are most likely to get onto the diseased condition. And that is what our main concern is. Okay. So with this particular data, we can do a plethora of things also, but I have already told you that is a subject of a project that you do, and how you can like come up with a mutant protein, and what is the relevance of that particular mutant protein, those we can find. But today I will just be telling you about how to screen for a particular thing. Okay, so how we can go about it. Okay, so you can go to simple search. You can also use the Alphamissense predictor. You can use it as a plugin for your any of the Chrome or any of the browser, or you can also use the large files from Zenodo to use it further. But we'll be talking about the web server based search. Okay, so you can go to search, and here you can search via multiple means though, so you can go to the question mark part. So what will work is UniProt accession ID will work, any entry name of the protein will also work, gene name will also work, or a ensemble transcript ID will also work. So those are the search terms. So this is what we call as search term. Basically, what are the things that you can search it using? Okay, so let's take an example of maybe BRCA1, BRCA1 for your breast cancer, or PNPLA3. Let's take PNPLA3, which basically causes your liver cancer. Okay, so not only liver cancer, fatty liver, then liver steatosis, and your fibrosis to then to liver cancer. Everything it is like having a role in. Okay, so let's try to understand that. Like likely benign, pathogenic, ambiguous, everything it will take. But apart from that, if you want to set a cutoff of the pathogenic score, that you only want the data for scores above that you can set, of course. Okay, lowest possible or the highest possible cutoff that you can set possible, or you can simply submit and see all of the possible results. Okay, so these are all of the possible results. Okay, it is having likely pathogenic, ambiguous, everything is there. Okay, so ambiguous means 50-50. Okay, likely benign means less than 50% of chances. Okay, of less than 57% to be precise, to get a disorder. And then you have likely pathogenic, basically, which is having more than your 57% of chances to get a disorder, whereas more than 80% are connotated by deeper shade of red, or you can say maroon. Okay, so this is likely pathogenic, which is having more than 80% of probability to be converted to a disordered condition. Here you can see what is the change. Okay, so here you can see ASP, third is the position. Okay, third position TRP. Okay, So it would be converted to TRP. Okay, so a better understanding you can get once you download the data. PNPLA3 human is the accession ID for Uniprot, and for Ensemble you have this particular accession ID. You can also cross check this particular thing. Okay, so I will make you cross check also. Okay, so if we can open the Uniprot and go via this particular ID, we should land up to the wild type of this particular thing, and like the position that you see should also follow. Because this is the same protein, don't get confused, these are the different position of amino acid of the same protein. So that is why the protein ID is not gonna change. It is for the same protein, this many variances are possible for each position, how many alterations are possible, okay, which are not yet been discovered. So it is not that all of the things that you see are being found in the world. Okay, so that is why it is a predicted thing. Okay, but the prediction nature is very high, so that is why we are counting upon this, because it is taking the data from ClinVar and Alphafold. Okay, so if I hit on download of the whole data, so that I can do, it is downloaded basically. I can also do a cutoff of basically only the likely pathogenic, because the ambiguous and benign I am not interested in. So it will give me only the filter out all of the else, and will give me the likely pathogenic results. Okay, here you can see the pathogenicity score will always lie beyond 57%. Okay, so you just do a multiplication into 100, it is percentage. So why the pathogenicity is like 0 point something, because I have already stated that probability lies always between 0 to 1. Okay, so that is why. So it is basically 59.9%. Okay, so likewise, so I have downloaded the data. I would open it using numbers. Okay, so this is the numbers. You can use, if you are using a Windows, you can always use Excel sheet for the same. So it will work the same. Okay, so likewise, okay, so here you can see this is the data. Okay, so how many entries are there? Okay, so I can also do a comparative analysis. By the way, I will open this particular Uniprot. In the Uniprot same ID I will open, and I will do a cross validation or cross check of the same. And you can observe a thing, that how much of a variance are there in the Uniprot. Okay, so which are not predicted, which are actual. Actual means which are being seen across the world. Okay, so this is PNPLA3 underscore human. You just copy it and paste it in here. Okay, or you can simply search via PNPLA3, still you will get the same result, by the way. Okay, yes, so see, it is here. So this is the ID that we were talking about. Okay, so if I go on to the sequence, 481 is the sequence, and each of the amino acid can change to anything else than the main one. Like M cannot be replaced by M, no. M can be replaced by all other sort of amino acid, and what would be the repercussions of those. It is not that all other 20, 21 or likewise amino acid it can change onto. It is just that what would be the relevant thing to do. So that is based on that it would be having the results. Okay. So if I go to variance viewer, you can see how many variants are been reported as of now. Only 697. Okay, 697 results that you can see. At the bottom you can see all of the results. Okay, so expand table, so you will be able to see for each positions what are the variances, which are the pathogenic ones, okay, which are the benign ones and likely significant. But these are not predicted results, these are directly taken from ClinVar. Okay, so it is a proven result. Okay, so wet lab based you can say, or basically worldwide procured data. Only 697 variances that are being enlisted over here. But for the same protein, okay, so for each variance, how many result, how much of a result this one is giving, Alphamissense. Okay, so let's take a look at that. Okay, so if we can do so this will make the count possible. Okay, so this is 9121. Okay, so if I remove the first, because the first row is of the the first one is for the template, your what do you say, for defining this particular thing. Okay, so still it is 9120 or approximately like that. Okay, So this much of a number, so like without screening likely benign, ambiguous and pathogenic. Okay, so it is having everything. Okay, but I can filter the pathogenicity based on the its percentage, and if I do so, so it will give me the highest percentage of pathogenics. Okay, so which we are interested in, because higher the level of pathogenicity, that means that particular variance will land up to a disordered condition. Okay, what sort of a disorder, you might ask? So that is not defined, but PN PLA3, whatever disorders PN PLA3 is involved in, it can be any disorder, not only the liver based disorder, any disorder that concerns to this particular gene, okay, or the gene is involved in that, disorder can be caused, okay, and it is having a very high probability of that. How can we cross check that? So let's cross check. So amino acid 1 is in the template protein, and amino acid 2 would be in the is the alternate amino acid. Okay, so just like you have your reference gene, reference allele and alternate allele, likewise, so this is in the template protein or the wild type protein, and this is the altered one. Okay, so Y is changed to A, then it is likely benign change. Y to C, likely benign. Okay, so Y to E ambiguous, whereas likewise for the second position, you can see only for the second position this many variances are possible. In the left side, it is always Y, because the wild type sequence is not going to change. Na? So the template sequence, for the template sequence for the second position, it will always and always be Y. Okay, we can cross check also. Let's look at this one. So this one is a wild type sequence only for the same protein. Let's cross check. What we were checking, second position Y. You can see second position Y only is there, MY. Second is Y. Okay, you can cross check at any point. Okay, so it will, the left side will always be resembling the wild type. Okay, 14th position G. Okay, 14th position, let's look at the L is 10, 11, 12, 13, 14. 14 is G. C. 14 is G, no? 14 is G. Likewise, you can cross validate every time it will resemble the wild type, the left side, amino acid 1, because that is the reference template allele. Not allele basically, because it is a protein, we have to call it as amino acid. Okay, so template amino acid. Okay, so allele we consider in the form of gene. Okay, so in genetics we call it as allele and all. So here it is template amino acid. So this particular amino acid can change to this, and what is the chances of this particular variance, or if this particular mutation happens at the human level, what are the chances that this particular variance might bring about a change in the protein structure and functionality, which will lead to a disordered condition? Not likely, because this is likely benign. Now if I change this particular pathogenicity score, because we have 9,000 and more entries, if I sort by descending order, if I put it, so now I have the highest pathogenicity score at first. Okay, so at 118th position, if the L, that is the in the wild type, okay, so you can cross check. So at the 118th, so this is 200, 118th, this one. Okay, K, no, it is L only, wait, 118, sorry, not 80. 118 would be somewhere here. 120, this is 118. Okay, so L. Okay, so here you can see 118. So this is L. So L, if the L changes to D, then it becomes very, very highly pathogenic, 99.91%. Okay, so almost 100%. If you can round it off, 100% chances that this particular variance will, basically in the protein, if only the at the 118th position, obviously all of the mutations are not gonna happen simultaneously or at once. Okay, so we are talking about in the whole protein, in the whole this particular sequence, the full sequence, only the L, only this particular L changes to D at the 118th position, that might bring about a change in the protein. Not might, basically 100% chances that that particular variance will lead to a disordered condition, because that would change the functionality of the protein for a devastating effect basically. Okay, that is why it will be highly pathogenic. So because I have used it from your ascending to descending, so of course the highest pathogenicity would be put into a chronological order that you see. Okay, Likewise, you can follow that. Okay, and based on this, we can infer which particular variance is going to cause a disorder, and likewise, and based on this, we can come up with a mutant protein also. We can do this particular mutation manually, and we can model that particular protein in Alphafold or ESM fold, bolts 2, or even the bioinformatics based tools like your FireRay or Swiss model or ITSR and this that. Okay, so like we can come up with a mutant protein which will cause a disorder, and using that particular protein further to come up with a drug molecule. In drug discovery also we can use that particular protein which like that particular mutant protein which has the potential of causing a disorder. Okay, so that was the main agenda of this. And if you intend to do a project on similar lines, so we have a whole lot of project basically. So this particular Bio-ProTech company, so you can like basically contact me via LinkedIn if you are willing to do a project on any of the things. We have a whole lot of project on cancer genomics, on your variance prediction, and like protein modeling, AI based things. Okay, so also I will share the Google form with you, which particular project you want to work with, and you can talk to me regarding the all the project regimes that we have and what sort of things that we provide. Okay, so with that note, so this is Pradyut Banerjee signing off. See you in the next video. Till then, keep practicing, and if you are new to the channel, please consider subscribing so that we can get a good reach and we can get started with the further part of the things and come up with more such projects, okay, in the future. Okay, so with that note, thank you everyone for watching. Have a nice day ahead.